Hi Tara, welcome to the pGLO lab. This video will show you how to conduct the first day of the lab, and it will detail what you and your students will need to conduct the lab. Let's start with what the lab looks like. This is my lab, and it's a bit busy, but you can see that there are several individual workstations. I have students work in groups of 3. What you can see here is I've prepared the tubes, plus and minus, that each contain the transformation solutions already on ice. Each station also has each of the components students will need in order to complete the lab. Notice how the plates are labeled. More about that in a minute. On another bench, we have our plasmid on ice, the water bath set to 42 degrees Celsius, and an incubator set to 37 degrees C. Behind these, I've just got extras of everything that the students might need for the lab. Now, how to label the plates. If you stack them like this, you can easily just run a marker up the side and it makes it easier to label your plates. 1 streak is for Luria broth plates, 2 for the LB amp, and 3 for LB amp era. The first step is for students to select one of the colonies from the starter plate you've prepared 24 hours in advance. Using an inoculating loop, the students will take the colony and then transfer that colony to one of the tubes containing the transformation solution. From there, the students will take a fresh inoculating loop, select a small amount of the plasmid DNA, and add it to the test tube with the plus. This tube contains the E. coli and transformation solution. Note the beaker to the side, which is there for easy disposal of used inoculating loops. The students will then incubate the tubes for 10 minutes on ice. During the 10-minute incubation period, students should label each of their plates appropriately. The students will then heat shock the E. coli cells by placing them into the 42 degrees Celsius water bath for 50 seconds. It is imperative that students do not go over this time. After heat shock, the tubes are incubated on ice for 2 minutes. Luria broth is then added to each of the tubes, and then the samples are then added and plated to the plates.